Have you ever gotten a paper cut? That sharp sting, the tiny line of red, and the way it makes you wince, it's a universal experience. It stings, but soon your skin repairs itself, almost like magic. Within days, the cut vanishes, leaving barely a trace. Our bodies are constantly at work, patching up the little damages of daily life. This ability is called regeneration, and it's one of nature's coolest tricks. From plants sprouting new leaves to animals healing wounds, regeneration is everywhere in the living world. Our bodies are always fixing themselves. Skin heals, bones mend, and even our liver can regrow. The liver, in fact, is the only human organ that can regenerate significant portions of itself after injury. But our healing powers have limits. Lose a finger, it's gone for good. We can't regrow entire limbs or organs the way some creatures can. Damage your spinal cord or your eyes' light-sensing cells? No natural fix. These injuries are permanent, and medicine can only do so much to help. The photoreceptors in our eyes, once lost, are gone forever, and vision suffers. These cells are essential for seeing, and their loss can mean blindness. Many things can damage these delicate cells: genetics, aging, or injury. Over time, millions of people lose their sight to these causes, with no way to reverse the damage. Now, meet the zebrafish, a tiny aquarium fish with a superpower. In labs around the world, scientists study this remarkable creature for its unique abilities. If its retina is damaged, it regrows the lost cells and restores vision in weeks. Salamanders regrow limbs, starfish regrow arms, and these feats seem almost like science fiction to us. And some flatworms can regrow their whole bodies. Cut one in half, and you get 2 living worms, each a perfect copy. Why can these animals regenerate, but we can't? The answer lies in our cells and their genetic programming. It's a story written in DNA, shaped by millions of years of evolution. As humans evolved, our cells became highly specialized for complex tasks, but we lost some ancient regenerative abilities. Our bodies traded flexibility for efficiency and complexity. Evolution made a trade-off: specialization over regeneration. This choice helped us survive, but it came at a cost. Zebrafish kept the genetic pathways for regrowing complex tissues. We mostly lost them. Their bodies can activate genes that ours have long since silenced. Understanding this difference is the first step to unlocking our own regenerative potential. If we can learn from these animals, we might one day heal injuries that are now permanent. Could we ever switch those powers back on? The answer may change the future of medicine and our own bodies forever. The secret is in our DNA. Hidden within our genetic code are the instructions that determine how our bodies grow, heal, and adapt to injury. But not all creatures follow the same script when it comes to repair. Zebrafish have special cells in their eyes, Mueller glial cells, ready to transform into new photoreceptors when needed. These tiny fish possess a remarkable ability to regenerate their retinas, restoring vision after damage that would leave humans blind. When the retina is injured, these cells revert to a stem cell-like state, divide and rebuild the lost neurons. It's like having a built-in repair team, always on standby, capable of replacing damaged parts with brand new ones. In humans, our Mueller glial cells don't regenerate. Instead, they form scars, a process called gliosis. This response is protective, but it comes at a cost. Scar tissue protects in the short term, but blocks new neuron growth, making vision loss permanent. Our eyes lose their ability to repair themselves, and the damage becomes irreversible. Why the difference? As mammals evolved, we prioritized stability over regeneration to avoid risks like cancer. Our bodies chose safety, trading away the power to regrow complex tissues for protection against runaway cell growth. Our Mueller cells are locked in scar mode, unable to rebuild from scratch. They're stuck in a defensive stance, unable to switch back to their regenerative roots. That's why diseases like macular degeneration or retinitis pigmentosa cause irreversible blindness. Once the retina is damaged, there's no natural way for our eyes to recover what's lost. Our repair crew can only patch, not rebuild. Imagine trying to fix a crumbling building with only duct tape and plywood, effective for a while but never truly restored. The challenge for scientists: can we unlock our Mueller cells' dormant regenerative abilities? If we could, it would revolutionize how we treat vision loss and other neurodegenerative diseases. If we could flip the right genetic switch, maybe we could turn scar formers into master builders. The tools of modern genetics, like CRISPR, offer hope that this dream could one day become reality. The answer could change the future of vision loss. Imagine a world where blindness is no longer permanent and sight can be restored. Can we find the key? The search continues, and the possibilities are as vast as our imagination. Here's where science gets exciting. Researchers in South Korea asked, can we make mammalian Muller glial cells act like zebrafishes? They studied mice whose Muller cells normally form scars after eye injury. The team suspected a specific protein was acting as a stop sign for regeneration. They blocked this protein in the Muller cells, and something incredible happened. Instead of forming scars, the cells began dividing and transforming into new photoreceptors. They'd flipped a genetic switch, coaxing the mouse eye to regenerate its own retina. This is proof that our regenerative potential isn't gone, just dormant. By targeting these molecular breaks, we might one day help human eyes heal themselves. It's a radical new approach to treating blindness. To truly appreciate this breakthrough, let's take a closer look at the retina, the eye's very own movie screen, where the magic of vision begins. Your eye works much like a sophisticated camera. The cornea and lens at the front focus incoming light, adjusting for distance and clarity. But it's the retina, a thin layer of tissue at the back of your eye, where the real story of sight unfolds. The retina is densely packed with millions of specialized cells called photoreceptors, rods which help you see in dim light and at night, and cones which allow you to perceive vibrant colors and fine details in daylight. When light enters your eye and strikes these photoreceptors, they convert it into tiny electrical signals. These signals are the first step in transforming light into the images you see. Without these photoreceptors, no matter how perfectly your eye focuses, there's nothing to actually catch the light, no way to start the process of seeing. The retina processes these electrical signals and sends them racing along the optic nerve to your brain, where they're assembled into the vivid, detailed world you experience every day. But here's the challenge. Because the retina is made of delicate nerve tissue, its cells don't naturally regenerate in humans if they're damaged or lost. Diseases like retinitis pigmentosa and macular degeneration slowly destroy these precious photoreceptors, leaving behind blank spots or dark patches in your vision that can't be repaired by glasses or surgery. As more and more of these cells die, vision becomes blurry, distorted, or even disappears entirely, leading to irreversible blindness for millions of people worldwide. That's why the idea of regrowing or replacing these cells has become a holy grail in the world of medicine and vision science. Now, thanks to groundbreaking new research, we're closer than ever to making this dream a reality, offering hope to those who once faced permanent vision loss. Think about how your skin heals after a cut. Blood clots, immune cells clean up, new cells rebuild the tissue. It's a coordinated dance perfected by evolution. Now imagine applying this to the eye. In zebrafish, Muller glial cells act as a reserve army. When photoreceptors are lost, they rebuild them perfectly. It's like reconstructing a computer chip so it works just like new. In humans, our Muller cells have lost the instructions. They only form scars, not new cells. The Korean scientists' breakthrough suggests the blueprints for regeneration are still in our DNA. The challenge is giving our cells access to those blueprints again. Regenerative medicine aims to upgrade our repair kit to match zebrafish. If we succeed, we could restore sight by rebuilding the retina from within. The potential is enormous. The future of healing could be transformative. So, who are Muller glial cells? In the retina, photoreceptors are the stars, but Muller cells are the essential support crew. They span the retina's thickness, connecting to nearly every neuron and providing structural support. They manage the chemical environment, clear waste, recycle neurotransmitters, and shuttle nutrients. In zebrafish, these cells can transform into stem-like cells after injury, dividing to create new neurons. In mammals, Muller cells do all the caretaking but lose the ability to regenerate. When faced with damage, they form a dense glial scar instead of rebuilding. This scar seals off the area but blocks new neuron growth. The difference between zebrafish and humans is in the genetic programming. If we can reawaken the regenerative side of Muller cells, we could revolutionize eye repair. They're the key to unlocking natural healing in the retina. The next step is learning how to flip their genetic switch. The Korean team's discovery centers on a protein called PROX1, a master switch in Muller cells. In healthy retinas, PROX1 keeps Muller cells in caretaker mode, preventing them from dividing or transforming. In zebrafish, injury drops PROX1 levels, unlocking regeneration. The researchers wondered, what if we lower PROX1 in mammalian Muller cells after injury? Using genetic tools, they blocked PROX1 in mice. The result, Muller cells began dividing and forming new photoreceptor-like cells. These new cells integrated into the damaged retina, expressing light-sensing proteins. By blocking a single protein, they reprogrammed Muller cells to regenerate. This could be the key to unlocking our own healing superpower. The implications for treating blindness are profound. The PROX1 discovery offers real hope for millions with incurable eye diseases. Conditions like retinitis pigmentosa and macular degeneration destroy photoreceptors, leading to blindness. Until now, medicine could only slow the damage, not reverse it. Imagine a treatment that coaxes Muller cells to regenerate lost rods and cones, repairing the retina from within. This could move us from managing disease to actually restoring vision. Of course, this research is in mice, and much work remains before human treatments. But the principle is revolutionary. Our eye's potential for regeneration can be unlocked. It's a new strategy, harnessing the body's own healing to fight blindness. Retinal regeneration isn't the only path to restoring sight. Science is exploring many frontiers. Bionic eyes and retinal implants use microchips to bypass damaged cells, offering artificial vision. Optogenetics delivers light-sensitive genes to surviving retinal cells, creating new artificial photoreceptors. Lasers and nanoparticles can stimulate retinal neurons, sending visual signals to the brain. These technologies are amazing, but often provide limited or altered vision. The beauty of regeneration is restoring the eye's original biological hardware. Imagine a treatment that lets your eye heal itself, regrowing natural high-resolution vision. The journey from lab discovery to human cure is long, but every breakthrough brings us closer. The ability to regenerate may be sleeping within our cells, waiting to be awakened. The future of sight has never looked brighter.